Good afternoon, everybody. I would like to welcome you here at the webinar called Introduction to High Throughput Screening. And in the first, I would like to ask you to turn off your cameras, okay? So all of you are muted, so you cannot speak during the webinar. And I would really need if you turn off your cameras, okay? Please do it right now. Turn off your camera, okay? Otherwise, you will be recorded and you will be on the recorded version of the webinar, okay? Hopefully everybody hears me, okay? So, my name is Sarkishimova and I'm from the Institute of Molecular Genetics in Prague, Czech Republic, and I will be the guide during the webinar. The main speakers who will be presenting the particular topics are Francisca Vicente from Medina from Granada in Spain, Sheraz Gul from Fraunhofer Institute for Molecular Biology and Applied Ecology in Hamburg, Germany, and Pepo Brea from University of Santiago de Compostela in Spain. This webinar is organized as the training activity of EU OPENSCREEN Drive project. EU OPENSCREEN is the European consortium of high-capacity screening platforms around Europe, and the DRIVE project is supporting the activity of EU OPENSCREEN partner sites and potential new members of EU OPENSCREEN consortium. On the slide, you can see the main topics of this webinar, ended with a short video of high-throughput screening process. You can write your questions during the webinar in the chat window. The questions will be answered either directly after the webinar or by email after the WebEx meeting is finished. Now, I would like to invite our first speaker, Francisca Vicente, who will start with target identification and definition. Then Pepo Brea will continue and talk about compound collection. The Sheraz. Can you turn off your... Okay, sorry for that. And Sheraz, who will talk about assay optimization, primer screen, and hit selection. Francisca Vicente will finish with data analysis, and then we will see the short video, which will be commented by Pepo, and the video was prepared in his place, in his partner site, University of Santiago de Compostela. I would like to ask Francisca, please. Francisca, can you start?
Yes. Okay, move to the first slide, please.
Yeah, I'm moving, but it's slow. Okay, good.
Okay. Hello, everyone. As Sarka mentioned before, in this course of the introduction of the my name is Francisca Vicente. I am responsible of the screening and target validation at the Fundación Medina. The first part of this HTS introduction course will be focused in the target identification and assay validation. For this purpose, the first thing that we are going to describe and the first things that we are going to be in detail is what is a target. The target is considered as any biological and any protein and any gene that could be used as the first tool to perform an assay and to describe an assay that we want to develop for the future campaign of screening. We consider a target that the target should be very important, like a protein, like a nucleic acid, carbohydrate, and lipid. That is very important to take in consideration that this target should be efficacious, safety, and meet the clinical needs when we think in develop an assay. And one of the most important thing to take in consideration is that this target should be degradable. What means degradable? Degradable means that this is accessible for the molecular or the extract of the drug that we are going to use in the screening campaign. And then this molecule or this strat or this drug at the end could be biological, could be a small molecular, but this should be bind with the target that we selected. And is the thing that we are going to see at the end of this target that we selected is a biological response. This biological response could be measured for different methods, but depending on the assay that we want to establish, this biological response should be evaluated by in vitro or by in vivo. Moving to the next slide, please. How to choose this target? Now, with these two tools that are described in the slide, we have a lot of knowledge about the genomics, the proteomics, and also we have a very important tool that is going to be involved the other two, the genomics and proteomics, that is the bioinformatics. With these three tools, we can select it, which all of the targets that we are going to use in the selection and to establish the assay development. The knowledge of the genomics of more of the cell lines and more of the microorganisms and the parasitics give us, and also later the proteomics, this was very important because open a window for more targets that we are using previously. Before, we are using a number of targets that were very, very, very short, but with these two kinds of these two tools, genomics and proteomics, the number of targets that we can use in the assay development is huge, and is more than the one that maybe sometimes we can reach. For this, in the next slide, please. Okay. For this, in the next slide, the first ones that we selected for the genomics, proteomics, and using the bioinformatics for this selection, we have to validate the target. To validate the target, we can use all of the tools that are described here, but this is a step that is extremely important when we want to develop an assay. And we have to lose in this step a lot of time. Studying the signal pathways of the target, studied if we have or is possible to have animal models for this target, to evaluate the chemicals and the genomics of this target, but at the same time, study all of the literature that is available about this target and also the patents. But if anything is not available in the literature and were not previously published, it's very important to work in this, using these tools, using immunochemistry, using Western plots, or using molecular pharmacology, in order to know before start the campaign of the screening everything about the target. This is one taking too much time, but this is going to give us a success of the campaigns of the screening that we are going to develop in the future. In the next slide is only a flavor of the main proteins family that had been used as a target. Sarka, I don't know if it's possible to put in a full screen the slides because the people is asking about this. Okay, no problem. Then some of these targets that are mentioned in these slides are know a lot for you because are GPCRs, are ion channels, alkinase, are nuclear receptors, are things that we are using many times. And in the top of the slide, you can see the percentage of where these targets have been using, and also the number of small molecules that were matched and were merged as activities of these targets. As you can see, there are ones that were used many times and were more easy to identify hits for these targets, but in other cases is more problematic. But this is only an example of the kind of targets. Thank you, Sarka. This is only an example of the target that we can use for develop the assays. And in the bottom of the slide is the percentage of the clinical success of these targets that had been obtained for many years, and with all of the family proteins that were described and mentioned before. This gives a flavor of one of the things that we can be used, but with the genomics and the proteomics science that are developing and technologies that are developing this, the number of targets that we can use and the families that we can use is increased a lot and is huge. This is a very good, this chart is from a very good review from Nature reviews in drug discovery, and it's something that we could be evaluated and read in order to know more about the different targets and how this could be useful. Move to the next slide, please. Now that we selected the target, we have to choose which one and to study all of the characteristics of the target. Now we are going to move other steps of the high throughput screening that is the first objective, is the outcome of the high throughput screening, is to select and to identify the hits. And also, I would like to mention briefly what some things about the leads. The leads is not the objective of this HTS, initial HTS courses, because it's one of the steps, less steps in the process of the high throughput screening. But I think it's very important to mention briefly this, what means leads and what is the difference between hits and leads. The hits is one activity that we selected to against the target that we desire and is acting against this target. The ideal is to obtain these hits a low concentration and with a lot of specificity. This could be sometimes probably, but in other cases it's only possible to obtain potent things. But we try to develop or to perform the assay in order to catch and to manage these low concentration activities. And one thing that we should consider is that is very important and is going to be described deeper later for my colleagues, is the importance to in the screen to try to use libraries with a huge diversity of compounds. This is give us more successful in the future screening. If the libraries that we are using doesn't have this don't have this diversity, the probability of success is less. And also the size of these libraries and the size of these campaigns is the numbers that you see in the bottom of the slides, but it depends of type of the slide of this assay that we are going to develop. It could be large, medium, or high, and this is very important. This is according also with the characteristics of the assay, the capacity of we have to do HTS, and the cost of the assay. But this is going to give you in detail later in the next steps of the courses. Please move to the next slide. Before to move to the rest of the presentation of this first part of the course, I would like to move very briefly of the terminology of the drug discovery in order to be all of us in the same picture and to be very clear this concept. These concepts are very important and everything should be understood in the same way. High throughput I mentioned before, what it means. Assay also it was mentioned very in detail. Is the assay is the thing that we develop with the biological system that we selected. The hit also is the outcome of that we obtain with the most of the screenings, and is the could be an inhibition or activation, it depends. And the next step that is new and is the first time that I mentioned is hit validation. Hit validation is the next step after we obtain we selected the activities of the screening, the primary screening that we are running. and is with the ones that we selected in the first primary screening, we need to validate it. And this validation is very extremely important, and is something that we should establish before start with the campaign of screening. Is the validation of if the hits that we obtain should be false positive, true positive, and it should be validated if they are specific or not specific. This is something that we should establish from the beginning. It's loss of the time if only we establish the primary screening and we don't consider this kind of secondary or this kind of screenings that gives us the functionality and the value of the hits that we obtain in the primary screening. And finally, briefly, to say you that the lead identification is the next step after the height validation. And for the lead identification, the hit that we are selected in the validation should be potent, should be specific, should be selective, and should be the one that we move later for the development programs. And this is the last step of our campaign of screening. But it's the objective of we have to think when we establish this process of development one assay. Moving to the next slide, please. Sorry. Next slide, please.
It's not working. Sorry, Francisca.
No worry. No, don't worry. We can wait.
Now?
Okay.
It's fine.
Okay. Thank you so much, Sarka. Now we are going first move in 3 slides. The first one is the hit discovery. That is the first step, and after is to mention the hit selection and the validation. The hit discovery, as mentioned before, is included the HTS screening that is the process that we are described previously, but now in this slide only is mentioned 2 points that were not described previously. How the screening paradigms and the strategies of the screening paradigms could be focus or unfocus. This means that focus is when we selected one library that is known, and we know which kind of components that we are going to work. And in the case of unfocus, we don't know any characteristics of the chemotype of the library that we are using, and then we can identify anything. According with the HTS screening that we are going to develop, in this case, it's possible to select it one focus strategic or unfocus strategic. But this is taking in consideration the account that we are going to select it in the phase. And also is taking in consideration if the target is completely new and then we don't have any experience about this target, and then we don't know any kind of pharmacophore that maybe could be the match or the merged the target that we identify. And in this case, it's completely unfocused. But if we have some experience and then we have some kind of the experience that of type of pharmacophores or the type of chemical library that we could be the ones and specific for this target is the one that we can select it. But this is also a very important point according with the kind of target that we are selecting. Moving to the next slide, please. Okay. These were mentioned before, but only to focus to mention 2 points here in the hit identification. The hit identification is the system that we develop and is the system that we selected for determining that the compounds interact with our target. And in this case, the system that we are selecting for this hit identification is very variable. There are later where you are going to see different methodologies, but these methodologies could be any molecular, any interaction, protein, any enzymatic protein, any readouts that give us this information, any expression of cell lines, and with this is very important for us and very significant, because if we selected very good these tools or this argument, these technologies, maybe to avoid later to use a lot of animals in the last step of the studies. And this is very important because it's safe, and you know that one of the ethical process of the drug discovery is to use less animal in the studies. That is something that we cannot avoid. We can use many technologies to avoid to use this proportion of animals, but at the end we need to do, before to go to the clinical trials, we need to do some experiments in the animals. In the next slide is the lead identification. I am not going to the characteristics that were mentioned before, but once that we have, I will be focused in these acronyms that is mentioned in this slide. That is, we selected the hit validation, and after we pass this to the lead, and should we have all of the characteristics that are mentioned in the top of the slide, but also should we have this lead this compound that is considered as leads, should we have ADMET properties. What means ADMET properties for the people that have not familiar with this? This is a terminology that is used a lot in the pharmacology, in the pharmacokinetics, and it's an acronym that means absorption, distribution, metabolism, and excretion. And when this is very important, and we should do very early this determine the characteristics of the hit that we are validated very early, and in a very early stage, because this gives us a lot of information that to avoid a lot of time and to avoid lost a lot of time, and to pass only to the lead optimization the best compounds with these characteristics. And to evaluate with these characteristics, we evaluated the side effects or the secondary effects that could have these leads later in the clinical trials. And to know this in the very early stage is very important, because we are going to have only to pass later in the lead optimization things that gives us very few problems. Okay, next slide, please. With this slide, is only summarize all of the things that we see. We see the target in the drug discovery, the 3 steps that were described previously: the target identification, the assay development, and the ASTS unvalidated. And the key questions in the assay development is the ones that we are described here. All of them should be considered. Should be considered if we are going to look an inhibitor and an activator, if the target could be regulated, if the target should be measured, because if we cannot measure the function of the target, we don't have any assay in our hands. To see if it's possible to do this process in ASTS, in a medium, large or medium ASTS, and also to pass to do the post-ASTS, that is the one that I mentioned before with the lead optimization. The next slide, please. And this is not the last, it's the previous one, but slides of this first part of the courses, but is a summary of the assay development, is the key consideration in the assay development. All of them were described before, and were mentioned and emphasized that they are very important. They are very important, robustness, reproducibility, but one of them were not mentioned before, and it's something that is very important, is the cost. The cost is very important to take in consideration, because not all of the assays could be run at the same in a low cost, but some ones are very expensive. But if the outcome that we are obtaining from them are very important in the evaluation of our target and in the evaluation of our leads, maybe we should consider this not with a very large amount of compounds, but with some of them very specific, because maybe is the test and it's the process that gives us the next step to pass this to the next trial or to the chemical trial or to the leaf optimization. But it's something that we should balance according with the results that we obtain. And I would like to finish with this quality, this sentence that is here, that is extremely important. To lose a lot of time to develop the asset is extremely important because this gives us later a very good results. And with these results, we could move forward. But if we do very quickly, we develop a very quickly asset and we don't obtain very robust results or quality data, we lost the time. We lost the reagents, we lost the time, and we don't have any answer in our hands. Okay, this next slide. The next slide is only a brief mention to say to you things that we should consider when we develop an assay, because there are things that are very obvious, but sometimes we don't take in consideration. And these kind of causes are the causes of more of the variation of our assays. You can read that there are things that maybe some of them are silly, but sometimes created us very important problems when we develop an assay. Could be in both type phases, but ones of them are very specific for the cell-based assays, but the other ones should be only common for both those type of assays. I think taking in consideration these very normal or very obvious things are very important when we develop an assay. And with this, I think is all of the things and the points that we want to cover with this target in the first part of the course, the target identification and assay development. And thank you so much for listening. And now I think we can move to the second part of the course. Thank you so much.
Okay, thank you, Francisca. So I would like to ask Pepo to continue. Pepo?
Thank you, Sarka. First of all, just a brief introduction. My name is Pepo Brea from the University of Santiago de Compostela, and I'm the head of the high throughput screening platform of Biopharma Research Group at this university. And what I'm going to speak to you about is about the compound collection relevance in high throughput screening. As Francisca has pointed out, it's critical to have a good assay developed for having a successful high throughput screening, but it's also as critical as the assay development, the chemical library you are going to screen. And this depends on the purpose of your screening. If you are looking for novel drugs, if you are thinking a drug discovery process, the composition of this chemical library would be different for those projects that are more devoted to novel chemical probes, chemical biology process, as they are those carried out by EU-OPENSCREEN. On the left of the slide, these are a brief summary of the properties of known drugs. And as you may realize, all of them share a molecular weight around 300 and 400 more Daltons, more or less, a CLOP around 4 to 5, around 5 rotatable bonds, 5 hydrogen acceptors. These are the averages. So if you are thinking in obtaining novel drugs for a high throughput screening, your chemical library should have numbers similar to that in the chemical description of your compounds. But instead, if you are looking for novel chemical probes, you can not be so strict with these rules, and you can include in your chemical library another kind of products, as they can be also natural products. On the right part of the screen is a comparison about drugs that are depicted in red and natural products libraries that are depicted in green. And you may realize that the properties are quite different. So depending on the purpose of your screen, you have to select the right chemical library you want to screen on that in order to have a successful high throughput screening. If you move to the next slide, please. It's also important to know the compounds that are composing your chemical library. For example, if there are toxic compounds there, if there are reactive compounds there, if there are unstable compounds there. So these criteria that appears in this table are the criteria that we apply for designing the EU OPENSCREEN chemical library, in order to avoid these compounds that may add interferences in the assay and that could render big numbers of false positives due to their reactive nature or due to that they are covalent binders and properties like that. You should not discard them if you are looking for chemical probes, but you should be aware of those compounds, having some kind of warning or a flag of this compound that if it is a hit, you could think, okay, maybe this could be due to these chemical properties. Of course, there are some other compounds that makes no sense to be included due because they are toxic for almost everything. But you have to select quite strictly the compounds you are going to include in your chemical library. If you move to the next slide, please. As Francisca pointed out, it's also important to have a big chemical diversity in your chemical library. And this chemical diversity will guarantee or will increase your probabilities of finding hits on when you are running your screening campaigns. But sometimes the cost is a limiting factor, and you are not able to screen the whole chemical library because of there's a costly reagent or something with a say is hampering to run high throughput screening. So sometimes you can make subsets of your chemical library. What is on the top left is the distribution of the chemical diversity of a chemical library in cyan color, and in green color are representative compounds for the whole chemical diversity. So with selecting just these representative compounds, you are screening almost the whole chemical diversity on this chemical library. And these compounds are clustered based on their similarity. And this is depicted on the top right part of the slide, where there are clusters with just one component. There are around 50 clusters, and most of the clusters have 8 compounds or 9 compounds or 10 compounds in there. So once you find a hit for the representative compound of each cluster, you can screen the diversity around those compounds belonging to this cluster. And in this way, you are exploring the whole chemical diversity, but with a more rational way for those costly assays. As Francisca has already pointed out, sometimes you are going to run a focus screen because you know something about the target. These are directed screens where you know some properties or some compounds. So it's always useful to have also your chemical library characterized in terms of the families or of targets that you may bind, as they can be GPCRs, kinases, enzymes, nuclear receptors. And in this way, if you are going to screen just a given target, you just screen this subset of your chemical library in order to be more efficient, taking into account the cost of screening so many compounds and the results you are going to obtain. If you move to the next slide, please. Another critical point is to know the characteristics of the compounds belonging to your chemical library. That means that if you have, as an example, autofluorescent compounds, then this is in the top picture, where that when you acetate with a blue light and they emitting a green light, if these compounds are autofluorescent, they will render false positives due to this color. So it's always useful to have these compounds characterized by means of a bioprofiling in order to detect which compounds are autofluorescent, which compounds may form aggregations, which compounds may precipitate at a given concentration, which compounds may interfere with ATP as an example, which is a cofactor in many assays. So if you have this bioprofiling at the beginning, once you find those compounds on your high throughput, identify which compounds are true positives and which compounds are false positives. As Francisca has already commented, if you run a secondary assay by employing a different technology, as for example, the C part of the picture, that is a luminescent assay, of course, these autofluorescent compounds are removed from there, so these false positives are clearly identified. But there's still some warnings with this aggregation of these component-specific compounds. So it's always good to have your chemical library bioprofile in order to properly know what are the characteristics of your compounds in there. If you move to the next slide. And last but not least is also to make a proper compound logistics. Usually the most common procedure to store a chemical library is keep the compounds frozen at -20 degrees in 384 well plates or 1536, depending on the throughput of your facility. But in this paper that is from more than 10 years ago, the author showed the difference between having the compounds just in a lidded plate in the top and in a sealed plate. When the different colors in blue means the quantity of water that is catching the DMSO. The compounds are dissolved at 10 millimolar in DMSO. DMSO is hygroscopic, so it's attracting water. And you may realize that in the corners of the plate, you can be modifying the percentage of water up to a 50%, 15%. So the quantity of compound there, the concentration has been changed, but also there could be some hydrolysis there, or there could be some precipitations. Instead, if you use a sealed plate, you can see that the moisture catching is quite homogeneous in the whole plate. Furthermore, you have to avoid the freeze-thaw cycles. As much times you thaw your plate, the more water is attracting the DMSO. So you can see in the bottom of the screen on the left, when the compounds were stored at 4 degrees, that the DMSO is frozen at this temperature. When you screen them by the 5th time, the purity of the compounds had decreased up to a 50%. And in the 6th time, up to more than a 60%. So this is critical because when you are finding some results there, you are not finding results on your compounds, you are finding results on your impurities. So it's better to keep the compounds even at room temperature storage, as on the right, but under inert conditions, as an example, a nitrogen current or some kind of environmental control that avoids attracting this water uptake. So with this is just to summarize you the relevance of having your proper compound chemical collection, but also managing them in a proper way. And we can move to the next speaker. Thank you very much.
Okay. Thank you, Peppo. May I ask Shiraz to continue? Please, Shiraz.
Okay, thank you very much, Saka. So my name is Shiraz Gul. I am from the Fraunhofer Institute of Molecular Biology and Ecology, and I am Head of Assay Development and Screening in Hamburg in Germany. So I will cover the main topics in terms of assay optimization, pilot, and primary screening. So we've already heard from the previous two speakers various assay types which are required for screening, and also the compound library compositions as well. In terms of the assay development, there are 3 types of assays which are often used for screening, and this includes phenotypic assays where we often do not know the molecular target which we are investigating, and then there are biochemical assays where we have the purified molecular target, and then a cell-based assay where it's a halfway house between a biochemical and a phenotypic assay, where we have some sort of reporter system, either, for example, luciferase reporter or GFP, or some antibody detection looking at the molecular target within the cell. Most of the assays that we use for screening are developed in microtiter plates. And in terms of screening, once we expose our assays to compounds and we find our primary hits, these we do not often know the mode of action, and this is all the deconvolution work that is done afterwards. And ideally, we want to find compounds which are genuine modulators of our target, which might be activators, inhibitors, allosteric binders, and so on. But a large proportion of compounds could also be acting via a non-specific way. For example, aggregators, they could be optically interfering. For example, absorbance-based assays are very prone to optical interference. Fluorescence-based assays are less prone to optical interference. And luciferase reporter assays are even less prone to optical interference. There could also be a possibility that there is some low-level contaminant within your compound, which is the reason why you are seeing some activity. And then when you do repeat studies with a fresh batch of compound, it could be that the activity disappears. And this might be down to the fact that the sample that you originally used in the screen contains some low-level contamination. Ultimately, we would have a good data set, which would be our final validated hits, where we can be confident that the compounds are acting in a desired manner on the target of interest, and then they will be further optimized to deliver a lead compound for protection via a patent, and then move into animal model studies. The next slide, please. Most of the assays that we develop for screening purposes are based in microtiter plates, and this could be in 96 well, 384 well, and 1536 well as well. And all of these formats are used routinely for biochemical and cell-based assays. For imaging-based assays, the microtiter plates often have a clear bottom, so you can actually use a microscope to image the cells through the plates. And if you are using an absorbance-based assay, that may well also use a plate with a clear bottom. For fluorescence-based assays, TR-FRET, AlphaScreen, luciferase reporter, you will use, say, a particular colored plate, white or black, and that doesn't necessarily need to be see-through from the bottom. The advantage of biochemical assays are that as you have the purified target in isolation, we know the absolute concentration of the target. We can dose in substrate at a particular concentration. We can actually do kinetics of substrate turnover and do quantitative studies, including potency determination of compounds. And as part of the workflow before we do screening is to ensure that the assay is tolerant to DMSO, as the compounds that we use for screening are often dissolved in DMSO. And as part of the final workflow is to check the day-to-day variability and plate-to-plate variability of the assay. The assay, in terms of a reference compound, which is used to benchmark an assay, that should be consistent within a day and also within a plate-to-plate basis. With a cell-based assay, as we often do not know how much protein target has been expressed, there is a limited number of mechanistic studies that we can do with a cell-based assays. However, both are complementary. Often with a drug discovery project, you would probably want to have a biochemical and cell-based assay in parallel, and then you can confirm the activities of the hits in both assays and understand the mode of action of the compounds. Once you have your assay, and which is robust and reliable, and you have a standard operating procedure, this is often done in a manual format or a semi-automated fashion. You can then transfer the assay to a high-throughput screening robot if you are screening lots of compounds. And once you've done the screen, your initial hits, which might be activators, inhibitors, you would then have to pick those compounds again and do confirmation studies. This is essentially a repeat of the primary screen, but only with the primary hits. And once you've got your final confirmed hits, depending on the assay that you've used in the screen, you will probably want to run a counter assay where you would try and determine and prove that the observation is not some sort of assay-specific artifact. For example, with absorbance-based assays, if a compound is very colored, that may well absorb some of the light from the assay and give a false positive. So a suitable counter assay needs to be developed, and all the confirmed hits should be screened in the counter assay to ensure that the activity that you are observing is real and not some artifact. And then final progression is to run some sort of secondary assay. This is often a cell-based assay, which is more physiologically relevant, but looking at the same activity of the target, but in a different system. And you would expect a real modulator of your target of interest should be active in the primary assay and also the secondary assay. Next slide, please. In terms of assays, I've already mentioned that there are 96-well assays, 384 and 1536. Each of those has its advantages and disadvantages, and there really is no right and wrong in terms of assay plate density. If your assay is low throughput and you are only screening a small number of compounds, then a 96-well assay is fine. If you are screening hundreds of thousands of compounds, then you might want to miniaturize the assay into 1536-well format. But the bulk of researchers involved in drug discovery, they tend to screen in 384-well format, because this seems to be a good compromise in terms of throughput, cycle time, and also the fact that you don't need to invest too much in terms of very sophisticated liquid handling and plate readers. And the table at the bottom shows the plate density, the number of plates, and the assay volume and the total reagent consumption for a 100,000 compound screen with no dead volume. And really, there are diminishing returns when you miniaturize the assay from 384 to 1536-well format, as the assay volume often goes down from 10 microliters to just under 10, but you can try and push it down to 5 microliters. So unless your reagent is very expensive, you might find that the 384-well assay format is a good compromise and where you will not use too much reagent. Next slide. This marker library screen, in terms of a pilot project, this is often only done when you are intending to do a screen of 50,000 or more compounds. If you are doing say a 5 or 10,000 compound screen, you may well just go straight into the screen without doing a pilot screen to validate the assay, because time is money, and it may well be quick enough just to screen the entire library if it's a few thousand compounds. However, when you are screening 50,000 or more compounds, it is prudent to take a subset of the library, typically 1%, and then screen that subset in duplicate to get an idea on the performance of the screen, the performance of the assay, and then if you need to make some adjustments, you can do that before you embark on the expensive screen itself. This slide shows a summary of a 250,000 compound screen that we eventually did. So we picked just over 1% of compounds, which was in this case 2,760 compounds, and we screened them in duplicate in our primary assay, and the frequency distribution plot on the top left at 10 micromolar is what you'd expect. Most of the compounds are largely inactive, which is the purple line, and our two controls, which are the DMSO, gave 0% inhibition, and our reference control, which is the light blue colors on the right-hand side, gives 100% inhibition. The frequency distribution plot is good. You can see there are telltale signs of some optical interference. There are many hits which are coming up with very large negative inhibition, and also some compounds which are giving very large positive inhibition as well. These are very likely to be artifacts of the system, but we will come on to that in the next few slides. And then you can see the frequency distribution plot on the left-hand side and the correlation between the two data sets. Looking at the data by eye, this is a very good correlation between the two data sets. There will always be some outliers, and this is typical of screening, but very good correlation. And you can again see the two tails, which are giving very large negative inhibition and also very large positive inhibition. The bottom left are the plates from the marker library screen, which was done in 384-well format, and the Z prime. For biochemical assays, we typically try and have a minimum of 0.6 Z prime. For cell-based assays, as they are often more tricky and more difficult and less sensitive, and the signal window is lower, we can aim for a minimum Z prime of 0.5. But this is something that you need to decide as a team, what is a suitable Z prime for progression of the project. And the table at the bottom right shows a detailed analysis of the marker library screen at different percentage cutoff for selecting apparent hits. And just to add some extra value, what we did here was we screened the marker library at 3 concentrations, 5 micromolar, 10 micromolar, and 20 micromolar. And you can clearly see as you increase the compound concentration, the number of hits go up. And as you increase the percentage cutoff, the number of hits go down. So this table was used to decide what concentration of compound to use in the screen, and also what could be a reasonable percentage cutoff. For most screening activities, we try and aim for a hit rate of roughly 1%. However, this is chosen and decided upon on a case-by-case basis with your whole team. So in this case, we decided to screen at 5 micromolar and try and aim for a 50% cutoff as an apparent hit. This still gives us a 3% hit rate, which is more than what we want to aim for, but it gives us enough chemical matter if we need to go back into and pick more hits. If some of the compounds fail to progress, we've got enough chemical matter as backup compounds. Next slide, please. Having done the primary screen from the marker library scaled up to 250,000 compounds, there are lots of ways in normalizing the data and deciding what should be categorized as a hit compound, as whatever you decide to do then will really dictate how the project progresses. If you only pick hits which are from one scaffold and then you confirm them, you really are limiting the progression of the project. So there are lots of ways in normalizing the data, and the simplest way is to just simply select a particular percentage threshold as a cutoff. However, this is really not the right way to do things. You really need to get medicinal chemistry input from your team, input from public databases, Chembl, PubChem, DrugBank, PubMed, and really annotate the hits that you have already. This can all be done in silico, and then decide which compounds to actually pick for your further studies. As if you cut corners here or do not a thorough job, that will really have a significant impact on downstream activities. And often what you want to do is have exemplars from different scaffolds. You want to make sure that the compounds that you pick and progress are from multiple chemotypes. That gives you enough space, as some of the chemotypes will not progress further through the drug discovery cascade, so you can deprioritize them and then prioritize other scaffolds. Next slide, please. So having done the 250,000 compound screen, we did our triaging and hit selection, and then we picked roughly 2,500 compounds. So we did aim for a 1% hit rate eventually for confirmation studies, but we were aware that we do have some backup hit compounds which we can pick quickly if none of these compounds which are shown on this slide actually progress. So you always have something as a backup strategy just in case that's needed. This is a duplicate study of the confirmation of the 2,500 hits that we picked, n equals 1 versus n equals 2. And all of these compounds gave in the primary screen a 50% cutoff, which are shown in the green box. And you can see there are a number of compounds which do not replicate. This is typical. Not every compound will give a reproducible activity of 100%. So there will always be some compounds which will not confirm. If you have a confirmation rate of 50% or above, that is acceptable. If you have a confirmation rate of 10 or 20%, this really indicates that the screen did not perform well. So this is the output from the confirmation screening, where we pick the hits and this reconfirm the activities, and the bulk of the activities are confirmed. Next slide, please. And then you would run a secondary assay just to check that the compounds are active in a secondary screen, and then you would do dose response studies at a range of concentrations and determine the absolute potency of the compounds. We know theoretically dose response curves should be nice and sigmoid. They should have a nice slope factor of 1. You should have a minimum and a maximum of between 0 and 100%. In reality, screening gives a very dirty output. Some compounds, as you can see on the bottom row on the right-hand side, the third compound from the left, that's giving a reasonable hill slope and a reasonable signal window. However, As you can see in the top row on the left-hand side, there are some compounds which will only give partial curves. If you have sophisticated software which can analyze dose responses and remove outliers, which you can see in the top left-hand curve, software is available which will automatically analyze dose response curves and give curve fitting and give estimates for IC50 determinations. All this is possible. You can do some of these activities in Excel or GraphPad Prism, and when you are dealing with high throughput screening on an industrial scale, where you have 500 or 1,000 or 2,000 dose response curves, fitting them manually is too time consuming. So next slide. And the ultimate endpoint for this project, where we have screened 250,000 compounds, we have a data package where we have done our primary screen, we've done our secondary screen and our confirmation and our dose response studies, and we have a data package of a few hundred compounds with nice looking dose response curve, and you can see some screenshots on the top right hand side. And this is our final validated hit list. These are compounds which we are confident are modulators of our target of interest, and the potencies are typically in the single-digit micromolar region. And this data package will ideally contain compounds from different scaffolds, and then the members of the team will pick up these compounds and then progress certain scaffolds in the hit lead stage, which will involve optimization of the potencies, reducing off-target effects, and so on, and other parameters which are typical, required for lead compounds. The next slide. So our final validated hit list will contain compounds where we have typically IC50s of less than 10 micromolar against our target of interest. As we can relatively easily get some selectivity profiling done, profiling a small number of compounds, maybe exemplars from each scaffold, in a panel of selectivity assays should be done to get an idea on selectivity. There are also what we call anti-targets, those drug targets which we should not inhibit because they are required for biological processes, and profiling exemplars in these anti-targets should also be done. Before we actually hand over our data package to our team members, we need to confirm that the activities are real, and this should be done from a fresh sample of compound, either purified from your stock, or you resynthesize, or you repurchase a fresh solid sample, and the compound activity should be retained. As you have got scaffolds, there should be some structure-activity relationship apparent from the analogs. There should be no obvious undesirable chemical features within the compounds, and some physical chemical properties should also be determined. And much of this can be done in silico methodology. Next slide. That's where I end, and then I will hand over to Peppo to continue.
Okay, thank you, Shiraz. Francisca, can I ask you to continue briefly with the data analysis? Thank you.
Okay. The last part of this introduction course is related to data analysis. There are some parameters that need to be evaluated, the quality control of the assay, and some of them are described here in this slide. To evaluate the control, the quality control is very important in the case of the analysis, to be sure that the results that we are obtaining and the data that we obtain are robust, and then we are obtain a very good outcome of the assay that we are performing. One of these parameters is the zeta factor. The zeta factor is measure the robustness of the assay, is measure this robustness plate by plate, and should be over 0.5 or higher. And if in this zeta factor is evaluated the standard deviation of the sample, the controls, the mean of the samples, and the mean of the control. This parameter, if is if it's not reach the higher of 0.5 for each plate, this means that we don't should consider this plate as valid of our campaign, and then we don't consider this, and we need to repeat this one. This is something that is very important when we evaluate it and we do a large campaign of screening. Another factor is the coefficient of the variation, and where we measure the dispersion of our assay and of the results or the hits of the results that we obtain in each plate and each campaign. These factors or these parameters should be evaluated plate per plate, intraplate, and interplate. And finally, the third quality parameter that is very important is to evaluate the signal background and the signal noise. And this is to measure if it strengthens our assay or if it's not strengthens our assay. These three ones always should be maintained in quality. If some of them is not worthy and is not working well, we should think that maybe happen some problem with our assay or with our plate, with the plate that where we obtain these results. And then we could not consider this one for our campaign, and we need to confirm or to repeat this plate. In the next slide is only going to explain some specific characteristics of the theta parameter. There are some advances in the theta parameter because it's taking plate per plate, and then we give us the noise or level for each plate. That is very good because it gives us an idea of all of the plates of the run that we are performed in one day could be considered, or only we consider some of the plates, but not for this run. And in this case, try to avoid the problems that we have if we consider all of the run as something that is completely good. But the disadvantage is that sometimes when we evaluated the plate, we evaluated all of the samples that are in the plate, and then we evaluated the strengths of the plate. And the strength of the plates sometimes give us the problems of the issues that we have some border effects. And this border effect is something that is very common in some assays. And this advance could be an advance because if we have this kind of problems, it's giving us that the robustness of our assay is not good. And then you have to evaluate this evaporation or this effect that we have in the borders. Or maybe we cannot if we cannot solve this, we cannot consider the samples that are in the border of the plates, and then we need to avoid this number of wells that are in the plates. Moving to the next slide, please. Now, before to establish the readout of the assays, we need to establish the layout. This means the layout of the plate is the definition how we want to distribute the samples in a plate. This is in a plate of 96, but could be in a plate of 384, like Sarah mentioned before, or even higher. And in each plate will be the controls, positive and negative controls, in this position that is described here, but also could be in the middle of the plate, could be in some of the columns, could be, we try to avoid the borders of the plate, because if we have some effect border, it is control should be here and then give us some problems when we evaluated all of the samples and when we determine the parameters that we mentioned before, like the zeta factor of the coefficient of variation. All of the ones that we define this plate layout, all of the data that we obtain from this plate layout are uploaded in the database. And at the same time, in the next slide, please, is described another topic, another point that is very important to take in consideration that is assay plate creation, which will merge the distribution of the sample in the plates with the information that we have of each sample, and we should merge this in an automated process. If there are different limbs that could be do this, also we can develop an internal program or internal process to do this. But this, when we run a big campaigns with a high number of samples, this is very useful and it's very important. This is in order to try to avoid SS files. SS files could be useful when the number of campaigns that you run is too small, but when you run more than 100,000 samples in a campaign and in different targets at the same time, useful to try to do this in an automated process and to merge the samples with the results in an automated process using different systems that are available in the market. And in this next slide is an descriptions that the things that you obtain from the different instruments where you do the readout of the samples, we call this raw data, and this raw data are transforming and loading in this data in different graphs or on different plots. Many of them were mentioned before, and you see this kind of graph and plots for CERAT, and the sample that is here in the right of the slide is part of representative of one plate where you see the values, the raw data values, and at the same time you can establish different colors, and these colors could give you the positives and the negatives, and this scale of colors give you which one, and it's more visual. And at the same time, like presented Seraph, this can be transformed in dosage response, or can be transformed in different curves, different dosage curves. And to obtain or to come do this in graphs and in plots is very important, because it gives you from the point of view, and the first view of one are the hits that you have, the selection of hits, and what are the other things for the large number of compounds that you are obtained that are not important, and this is a selection. Also, there are many tools to do this, and some are free, and others are different process to do this in an automatic system. The automatic systems avoid errors and avoid to have maybe some results that at the end are not a good one, and the only thing that you are is to lose time and to identify some hits that are maybe not the right one. Next slide, please. This is my final slide before we pass to the next step of the courses. Is in this slide, I would like to summarize more or less the key factors to have a success of the ASTS. There are three main topics that we mentioned from all of the ASTS course. One is the quality. It's very important to maintain the quality of the ASTS process. It's very important to take in consideration, to take in account the time in the wells, the time in the DELs, in the screening, in the project, and also the last part on the last leg of this success, to obtain the success of this ASTS is the cost. Also, this topic was mentioned by Seraph in the presentation, and in taking in consideration the type of screening that we are using, this is more important or less important. With all of this that we presented, I think, and with these 2-3 point factors, we give you general presentation and a clear idea of some of the critical tools that can be used in the HTS strategies that give us the possibility to obtain some successful results to detect drugs that we could use later for the treatment of the important disease and obtain like the results of one of the current affecting to the whole of the world that we can suffer. This is very important to learn these things that we are trying to mention during this course and very useful for the success of all of the exits of the science in general. And with this, I finish the presentation.
Okay, thank you, Francisca. I will try to run the video. Okay, I'm able to run the video, and I would like to ask Beppo to comment the video which was prepared in his site. Beppo, please.
Thank you, Sarka. This video is just a brief illustration of how high-throughput screening campaigns are run. So in this video, you can see how the compounds are already prepared, and they are diluted in order to obtain the desired concentration for running the campaign. With this dilution, it is better always to have a single step where you can add all the reagents needed, if it's possible, to the compounds, and they are then moved to a liquid handling system that is going to add the compounds onto the cells from where we are going to measure the effect of the compounds. So the compounds are moved to the liquid handler, and everything, as you may see, is fully automated in order to be processing several plates at a time, and the cells are taken out from the incubator, and the robotic arm is going to take the plate with the cells from this incubator and move it to the liquid handler. So once the cells are on the liquid handler takes out the lid of the plate, and once the lid is removed, it's able to aspirate the compounds from the source plate and dispense the compounds into the cells. As you may see, all the plates are barcode labeled in order that the system is able to follow the process for each of the plates in this assay, and at the end, all the readout that you are obtaining at the reader is classified according to this barcode. So now, once the compounds were added into the cells, the robotic arm is going to remove the compounds from the liquid handler and return it to the source where these compounds were located, and then take the cells from the liquid handler and put them into the incubator, where they are going to be incubated for the required time and at the required temperature, depending on the assay you have previously developed, in order to observe the effect of the compounds on the plate. As you may see, the robotic arm is moving all the plate with the cells up to the incubator that is at 37 degrees in this case and 5% CO2. As everything is fully automated, several assay plates are running parallel. Each of these blue bars are belonging to a given plate. So the system escalates them in time in order to avoid that any plate is incubating for a longer time than any other. And once the incubation time is finished, the robotic arm introduces the plate into the reader. The reader reads the barcode, and the readout is carried out, and all the data obtained is fully stored in a file format that can be easily integrated into the databases, as Francisca showed, and from there you can easily review and obtain those response curves, and review those response curves, validate the compounds, etc. So this is just a brief summary of how the high-throughput screening campaigns are run Of course, it is something a little bit more complicated, but it's just to give you a brief flavor of how are these campaigns run. So with this, I finish. Saka, thank you very much.
Thank you, Pepo. Now I will stop the recording of the webinar, and we can continue with some questions.